Good morning. It's my great pleasure to introduce my longtime colleague, Dr. Julia Davydova, MD, PhD. Let me introduce her. Actually, she is from Tomsk, Russia. And she has graduated from medical school in 1993. And after that, she got PhD. Then came to the United States the year 2000. I invited her at the time. Then since then, we have been, they're working together. And so now, she is the assistant professor. And one of the milestones she has achieved is her own R01, on which I don't have any effort. That's completely her R01. So then I think, this title represents actually some of the content of your R01 stuff, right? So I'm looking forward to hear the recent outcome of her study, and please, Dr. Davydova, podium is yours.
Yeah, thank you. So thanks a lot. So can you hear me? Is that okay? Yeah. Thank you very much, Dr. Yamamoto, for introduction. Yes. So I have, as you could hear, I have joined the Gene Therapy Center at Alabama, the University of Alabama, at Birmingham in 2000. And since then, oncolytic virus therapy has become my passion, the focus of my work, my life. And we can see quite a few applications of viruses in the medical field. Vaccination, and of course viruses are very attractive vectors for gene delivery, and we can use virus-based vectors to treat non-cancer diseases. And of course we use viruses as oncolytics. So why viruses? I believe that virus-based therapeutics are the most interesting therapeutics on the current market. So those therapeutics are alive. Those therapeutics can multiply once they are inside of the body. Viruses are very smart. They're so smart that they can outsmart our immune system. They can use our own cells for their own needs. And in fact, some of them are considered to be the most devastating diseases to ever occur in human history. For example, smallpox virus. It's one of the main reasons for the decline of both the Aztec Empire and the Inca population. So this virus shaped and destroyed the whole civilization. This virus didn't spare the royalties. We can see that Egyptian pharaoh Ramses V has died in 1145 BC from this virus disease. And this virus killed Russian Tsar Peter II in 1730. And it's been estimated that smallpox killed around 300, probably even more, people just in the 20th century alone. So that's more than all wars combined. On the other hand, a very similar virus, the smallpox of the cow, or the cowpox, or variola vaccina, saved million and million lives. And that was English Dr. Edward Jenner, who in 1796 published his paper with a very long title, Inquiry into Variola Vaccina known as the cowpox, where he described the protective effect of cowpox against smallpox. And smallpox has been completely eradicated in 1980. And Edward Jenner's work they say that it saved more lives than the work of any other human. So it means we can use the viruses if we use them smart. And the concept behind the use of viruses for cancer treatment, in my opinion, is very straightforward. It's very logical. It's very rational. First of all, viruses can kill the cancer cells as a part of natural lytic cycle. So they can take a control of infected cells, and they can commandeer them as virus factories. And secondly, for million and million years, viruses have been transferring their genes into different type of cells, mammalian, human, plant. So they're naturally made gene delivery devices. Once again, I would like to underline that they're perfect if we would like to express some gene of interest. And surprisingly, that's not a new idea. People have noticed that influenza is being associated with tumor remission in the mid of 19th century. And by the end of 19th century, so we started actively employ the fluid from infected people to treat some cancer diseases. But the official discovery of virus happened later in 1892, and that was a Russian scientist, Dmitry Ivanovsky, who discovered a small pathogen while working with tobacco mosaic disease. And this pathogen was small enough to be trapped by the ceramic filter he normally used to trap bacteria. And these days considered to be the beginning of virology and virotherapy. So the first clinical trials with oncolytic viruses took place in 1950-1960. And at that time, that was a number of bizarre experiments from the current point of view, where hundreds and hundreds of patients have been treated with impure virus lysates. At that time, we had no idea how to purify the virus, so that's why the dirty lysates were injected into people, sometimes even infected body fluids. And it's exactly the case when in 1956 wild-type adenovirus was used to treat women with cervical cancer. Most of the viral particles have been arrested by immune system. So, and that was not any obvious therapeutic effect, but in some patients where infection took place, tumor remission was observed, and surprisingly, that was no toxicity. I can't help myself but mention that majority of these clinical trials were conducted by Chester Southam, a very talented oncologist from Masonic Sloan Kettering Cancer Center, who teamed up with Alice Moore to investigate the therapeutic effects of the first oncolytic viruses. And now he's considered to be rather a very grim figure in the field of cancer research. But at that time, being the leading figures, they absolutely contributed a lot into development of the oncolytic virus therapy. For example, they used the Egypt 101 extract from West Nile virus in more than 150 clinical trials. But at some point, Dr. Southam abandoned the use of mice and began transplanting human tumor cell lines, including HeLa, into human volunteers before treating them with oncolytic viruses. And this story has been very well described in this wonderful book, The Immortal Life of Henrietta Lacks. And of course, these experiments received harsh criticism. And even though Chester Southam was re-elected as a president of the American Association of Cancer Research 1 year after, I believe that actually it resulted in the punishment of the entire field of oncolytic virus therapy. But now we live in a different era. So that's the era when we can genetically modify our viruses. That's the era when we know everything, almost everything about the virus genome, And of course, we can make them safe. And there are different viruses, or let's say virus-based vectors we can use in the field of cancer research or in the field of gene therapy. That's, of course, retrovirus vectors, measles, lentivirus, the famous vaccinia, so which is very much popular right now. So adeno-associated viruses, adenovirus, and the herpes simplex virus, which was recently approved by FDA as the very first oncolytic virus therapy here in the US. I will be more focused on adenovirus because that's the scope of our work. So, human adenovirus. This virus was isolated from adenoid tissues in 1953. So, it's not associated to any severe disease. It causes a common cold. And since then, all the structural proteins of adenovirus, the entire replication machinery have been studied and scrutinized as well as the scientists can do. So we literally know everything about adenovirus and its genome, which is really good. So that's the non-enveloped linear double-stranded DNA virus. It infects divided and non-dividing cells. It possesses a very high transduction efficiency in vivo. So it does not rely on cellular death pathways as RNA viruses, which is also very good. It doesn't integrate into cell genome. It just hits and runs. And we do know that it's considered to be immunogenic. So, but on the other hand, adenovirus itself can stimulate a very robust anti-tumor immunity against cancer cells, which is also very handy. And adenovirus genome is perfect for cloning manipulation. So it has a great cloning capacity. So that's why we can easily insert the genes of interest. It could be the therapeutic genes, the suicidal genes, for example, or the imaging genes. And the very first selective adenovirus, or conditionally replicative adenovirus, we call them CRAD, was invented in 1992 by a small company called Onyx and by Dr. McCormick. And originally, this virus was designed to replicate only in p53 mutated cells. Later, we learned that this virus is not quite specific, and the deletion of the mutation of even B55 gene is not strictly related to p53 mutated cells. But at that time, that was a magic bullet. So you can just imagine the excitement of the health providers and the scientific community when such the ideal cancer treatment show up on the market. And of course, hundreds of clinical trials have been launched. But most of them showed somewhat disappointed results. The therapy was not effective when it was used as a monotherapy. And in 2003, this project was dropped by Onyx. And in 2005, Shanghai Sunway Biotech bought the worldwide rights to Onyx technology from Onyx. And a bit later, this virus was renamed, and it was approved for therapy of hand and neck cancer in China. So why we didn't see a bigger flash for actually this wonderful therapeutic and for the first generation of oncolytic adenoviruses? In theory, even one viral particle can kill the entire tumor mass as long as it can efficiently replicate and spread within the tumors. However, it's never the case, even in vivo models. And the experience from the first clinical trials with oncolytic adenovirus demonstrated a very poor spread of adenovirus in solid tumors. And as a result, a very limited anti-tumor effect. And now we know the reason. Most of the cancer cells, it's one of the reasons actually, are profoundly deficient for adenovirus primary receptor, Coxsackie adenovirus receptor. These clinical trials have also demonstrated the inability to treat systemic disease. So that's why we see a low enthusiasm among biopharmaceutical companies. On the other hand, all these clinical trials, hundreds and hundreds of them, demonstrated a good tolerance and safety. And speaking of which, I think that's a very controversial topic when it comes to the safety of adenovirus-based vectors. And I've been working with adenoviruses for 17 years by now. And I strongly believe that the safety is not the main concern of adenovirus-based vectors. And in fact, these viruses have a number of great characteristics in terms of safety. So as I mentioned, adenovirus is not associated to severe disease. It causes a common cold. So adenovirus possess an intrinsic selectivity for replication within cancer cells. Whenever you infect, for example, the normal human cells like the fibroblasts or epithelial cells, right, and in parallel cancer cells, of course you can see a much better replication in carcinogenic cells rather than in normal. Adenovirus does not possess any risk of insertional mutagenesis, and as I mentioned before, clinical trials with the replication-competent adenovirus demonstrated a good safety. However, the major limitation exists. We all know that adenovirus is immunogenic, so it can result into immune response. And subsequent to IV injections, most of the adenoviral particles target the liver. At least that's what we see in the mouse model, in the rodent model. Whenever we inject adenovirus systemically, maybe like 80–90% of viral particles go to the liver. And again, I can't help myself but mention the echo from the past. That's a Jess Gelsinger case. When the patient has died while undergoing the therapy with the adenovirus-based vehicle, that was the very first generation of adenovirus, replication deficient adenovirus. So, and his death is thought to be a result of a severe immunocytokine storm to adenovirus and the liver failure. And later on, the investigation committee agreed that was rather a very abnormal case. So they also found a number of fundamental errors in the clinical protocol, including a very high dose of adenovirus, a very poor condition of the patient liver, so that was a very wrong target. I honestly think that it has something to do with the quality of virus itself. So, but in any way, this tragic case negatively affected the whole field of adenovirus-based therapy and gene therapy, and created this kind of negative stigma about hepatotoxicity of adenovirus. So once again, I would like to underline that the vectors we work now are very different. So they're very different, so, they're more sophisticated and they're replication competent. So, and what are our strategies we use in my lab to improve oncolytic virus therapy? First of all, we can make the replication of adenoviruses specific, and there are many different ways. So the most popular way, so in our approach, is controlling the E1 gene, which is responsible for adenovirus replication with a tumor-specific promoter. So, and I mentioned already that most of the cancer cells are deficient for a Coxsackie adenovirus receptor, for the primary adenovirus receptor. So what we can do, we can switch adenovirus tropism and make our adenoviruses bind to other receptors than CAR. So for example, we can switch the adenovirus fiber knob from the virus type 5 with the knob taken from a different serotype, serotype 3, which binds to human CD46 and human desmoglin receptors, which are highly expressed on the surface of cancer cells. Or we can incorporate the RGT motif and make our viruses bind to integrin receptors, alpha V, beta 1, beta 3 receptors. We can express the adenoviral death protein, which is responsible for oncolysis and spread. We can retarget our adenoviruses from the liver by modifying adenoviral hexone protein, and it's been shown that that's adenoviral hexone protein which is responsible for binding to liver macrophages and blood factor 9 and 10. So the drawback of this hexone modification is that those viruses result in a very low yield, so making the productive cost quite unacceptable. Of course, we can equip our virus with a powerful therapeutic and or imaging transgene. And a few years ago, we created a system, we call it the Delta E3 system, so which allows to express the gene of interest specifically in tumor site at high quantities. So, and we can, you can see that we created quite more than a few vectors expressing different therapeutic and imaging genes, including GFP, luciferase, RFP monomer and dimer, SSTR, interferon, different interferons, interferon hamster and human interferon, sodium iodide importer, and now we are working on the colonial vector expressing cytokines to treat melanoma and sarcoma patients. And this system is based, as I mentioned, on this on the incorporation of all these genes into the adenoviral E3 region. When you insert the therapeutic gene into adenoviral E3, it's being controlled by the major relate promoter of adenovirus, which is active only when adenovirus replicating. So it means that expression from the E3 is replication dependent, and we can see the increased level of gene expression with each round of adenovirus replication, which is great. This structure is also known to overexpress the adenoviral death protein, so thus improving spread of the virus. And here we analyze the ability of these vectors to express transgene in target tumors, so upon IT, intratumoral injections, and upon IV injections. So, and of course, we analyze the ability of this system to monitor non-invasively intraperitoneal metastasis, and it worked also well. Here I would like to give a little bit more details about our project with a scope on radioiodine-based imaging and therapy for pancreatic cancer. So, and that would be about adenovirus expressing this wonderful dual function gene called sodium iodide importer, or NIS. So, what is the sodium iodide importer protein? That's a normal protein which is expressed in our thyroid and which is responsible for uptake of iodine. And its natural localization has been used as a way to achieve radiotherapy and imaging of thyroid cancer. And this therapy is being safe and effective for more than 50 years. People don't die of thyroid cancer here. So, and the absolute beauty of this protein is that it can work with different radio tracers and different radioisotopes, iodine-123, 124, 125, rhenium, technetium, and etc. And our hypothesis was to express this protein in other tumors than thyroid. So we could facilitate the imaging with, for example, iodine-123 using the SPECT-CT system, or imaging with iodine-124 when we use PET. And then later we could facilitate the iodine therapy with the therapeutic dose of iodine-131. And here it's how it works, and that's the phase 1 clinical trial of NS-mediated gene therapy for men with prostate cancer. And this clinical trial is being conducted by our collaborator, Dr. John Morris in Mayo Clinic. And that was John Morris who brought this protein and this approach to the field of cancer therapy. So that was him who established that. And I would like to underline that here he is using a very first generation of adenovirus, which was not produced in our lab. So that's the commercially available adenovirus at this time now. So that's the replication deficient, not specific. This expression is controlled by the Uvicta CMV promoter. So, and here you can see the a very strong spexity image of iodine-123 uptake in the prostate after a single dose of adenovirus expressing NIS. And later, this patient has responded well to the therapy with iodine-131. So, and we decided to improve the imaging potential and the therapeutic potential of this approach by creating our conditionally replicative adenoviruses. And we are using this virus at CMV-NIS as a positive control. And we made more than a few different NS-expressing viruses to optimize their structure with or without ADP, with different fiber modifications, and etc. And here we can see that, for example, this adenovirus, the best of them, can kill very aggressive pancreatic adenocarcinoma cell lines in vitro with such a low titer as 1 VP per cell. And of course, we analyze the selectivity of these vectors, and here we can see that COX-2 promoter control adenovirus expressing NIS shows no replication in normal human pancreatic tissues. That's the ex vivo experiment when we produce the tissue slices with our tissue slicer. So at the same time, we can see a very robust replication in adenocarcinoma tissues, and that's comparable to that of non-selective control. And we also showed that deletion of adenoviral death protein from adenoviral genome improves the sodium iodine symporter membrane localization, which is very important. So, and in fact, when we used this ADP plus adenoviral death protein plus and ADP deleted adenoviruses to track pancreatic cancer in vivo, we can see a great superiority of our ADP minus vector over the CMV-NIS positive control. It's almost nothing, and over the ADP positive counterpart. And we also estimated and evaluated this system in patient-derived xenografts. And once again, we can see a very strong imaging. I believe that's a great potential to track pancreatic adenocarcinoma lesions after a single injection of adenovirus, and compare it to this positive control, which is currently being employed in a phase 1 clinical trial. It's so much better. So, and we were very much happy to see that ADP deletion did not significantly alter the anti-tumor effect of our adenoviruses. So, but definitely it improved the radiotracer uptake, and that's the quantitative measurement of technetium uptake in patient-derived xenograft models. And we demonstrated a very strong oncolysis after infection with both ADP plus and ADP minus vectors. That was not that much with the infection done with positive control at CMV-NIS. And hematoxylin and eosin staining correlated very well with immunostaining of PDX tissues. here we can see a very active replication after infection with ADP plus and ADP minus counterparts. So, and green, that's the adenoviral hexon, and red is replication-dependent NIS expression. So, and of course, we demonstrated the ability of our system to facilitate iodine therapy with radioactive iodine when we just combine this virus with the therapeutic dose of iodine-131. And once again, so we could see the higher uptake of iodine in cancer tissues after infection with the no-ADP version of our adenovirus. So, but of course, our ultimate goal would be to move this approach to the phase 1 clinical trial. And that's the design of phase 1 clinical trial. We will hopefully open maybe in 3-4 years from now. And right now we are working on the IND protocol and setting up the pre-IND meeting with FDA. So, but in this trial, patients with locally advanced pancreatic adenocarcinoma will receive a single injection of NS-expressing adenovirus, and we will go with no ADP NS-expressing adenovirus via, and that will be EUS-guided intratumoral injection. And 3 days after, the NIS uptake will be quantitated with iodine-123, and radioiodine dosimetry will be determined based on iodine-123 uptake. And on the next day, the patients will receive an oral dose of iodine-131. And of course, patients will be monitored for the adverse effect of the virus and radioiodine. And I was very lucky to assemble a very productive and strong team of clinical investigators, including Edward Greena and Dr. Taylor and Brett Humphrey and Susan Casey from the radiology department. So, and hopefully, and we are very excited about this clinical trial. And I also believe that if this trial is successful, then this study will open avenue for use of radioiodine for other tumors than pancreatic cancer. For example, breast cancer is a very interesting target, or prostate cancer. So a few words about other project, and that's the collaboration with Dr. Yamamoto. That's adenovirus-based interferon therapy for pancreatic cancer. And I have taken this picture at the PanCan Purple Rides in Maple Grove this year. And I was very much overwhelmed to hear all the story from the pancreatic cancer survivors. And all those people are all pancreatic cancer survivors. And one of them is our friend who experienced this disease 12 years ago. And he's a great fan of interferon-based therapy because he strongly believed that that was this clinical trial, which was conducted here in the University of Minnesota, which saved his life. He's cancer-free for 12 years, and he's a very active pancreatic cancer advocate. And once again, he's a great fan of interferon-based therapy. And in fact, interferon-based chemoradiation therapy appeared as a very promising tool for pancreatic cancer patients. But despite all this incredibly improved survival rate of 55%, this approach has a number of limitations. We all know that interferon can cause severe systemic toxicity. Not all patients can tolerate these regimens. And also pharmacodynamic studies have demonstrated insufficient delivery and unstable level of interferon in tumor site because of short half-life of interferon. So our hypothesis was to express this interferon with our vector and then later combine it with chemo and radiotherapy, because interferon is known to be as a great chemo and radiotherapy sensitizer. Plus, interferon can stimulate immunity against pancreatic cancer cells. Just one slide here. Here we can see the therapeutic efficacy and survival rate of triple combination therapy in immunocompetent hamster model. So why hamster? Because they can permit the human adenovirus replication. They're immunocompetent. Plus, we have syngenic hamster pancreatic cancer cells, which is great. So we established the tumors and we treated with either adenovirus alone or with in combination with 5FU and radiation. And here we can see a remarkable tumor regression when radiation and 5FU were joined with adenovirus-mediated therapy. So, and the green curve represents the standard approach, which is currently being used to treat PanCan patients, 5FU plus radiation. So it didn't help at all. Radiation alone didn't help at all. And the plot on the right demonstrates the survival rate. And once again, the significant improvement was observed only when 5FU and or radiation were combined with the interferon-expressing adenovirus. So, and a few more words about the in vivo models. So, how much time I have, Masata? Okay, yeah, okay, good. So, the in vivo models, that's, I believe that's a very important topic. So, and the problem is that we don't have that many in vivo models to study safety of our adenovirus-based vectors. And the reason is that human adenovirus is very species-specific. And of course, that's a very normal thing for adenovirus to replicate only in human cells. It can, in fact, it can transduce, let's say, non-human cell lines, but it cannot produce viral progenies in most of the non-human cell lines. And historically, we are using two in vivo models to study safety and efficacy of our vectors. Of course, that's the immunodeficient mouse model, the immunodeficient mice bearing human xenografts. And we use this model to study efficacy of our vectors in human cancer xenografts. The drawback, adenovirus does not permit in mice. It means it doesn't make any sense to analyze biodistribution or safety of adenovirus in mice, especially if we are talking about replication-competent adenoviruses. And of course, it's impossible to study a host immune response in an immunodeficient model. We are very lucky to have Syrian hamsters, which are known to permit at some level replication of human adenovirus. And we are even more likely to have syngenic hamster cancer cell lines. So we have pancreatic cancer cell lines. Hopefully we will have melanoma and sarcoma cell lines, and we also have breast cancer cell lines from hamsters. So the dark side of this model, the immunological tools are not available. And of course, the main disadvantage of this model is that fact that hamsters and all rodents are not suitable to study efficacy and safety of our infectivity improved or tropism modified adenoviral vectors. Those vectors which are based on adenovirus type 3, which utilize adenovirus-free receptor. And now this receptor has been identified, it's CD46 and human desmoglein 2. So, and those receptors are not expressed in rodent cell lines. So what do we do? How do we study the efficacy of our vectors? And actually, most of those vectors I presented earlier today, they are based on this Ed3 serotype, and we are about to move them towards the clinical trials. And that's how we initiated the search. And we tested many different cell lines, including murine, rat, hamster, bovine, canine, and swine. And we could see that only porcine cell lines, so only pig cell lines, can be supportive for both, for binding and replication of our Ed5, Ed3 retargeted adenoviral vectors. And just a brief example here, we can see good signs of adenovirus replication. That's a staining of adenoviral hexone protein in PK15 swine cell line and in PTK75 cell line. And we can see the expression of NIS after infection with our COX-2 promoter control NS expressing adenovirus. And of course, we were wondering if we can use a pig as a host for Ed5, Ed3 modified vectors. So, and we initiated this study. That's the experimental design. And at all these time points, we collected liver biopsy and blood and urine and saliva. And at day 7, we harvested the swine organs, and the necropsy analysis and histology analysis have been performed by certified vet pathologists. And everything was done with the help of experimental surgical services directed by Dick Bianca at the surgery department. And here I just summarize some of this data. That was no adverse effect on hematology parameters. The red blood cell count, white blood cell count, hemoglobin was normal, and platelet count was normal. And here I would like to clarify that in a mouse model, whenever we inject adenovirus systemically, it results in a very robust thrombocytopenia as a result of activation of complement and toll-like receptor pathways. We don't see it here. We don't see it in pigs. There was no major effects on serum chemistry. So the ALT, the liver-specific marker, was normal. AST, LDH markers were normal. That was some elevation of SDH after infection with Ed5 fiber, but still it was within the normal range. And we could see the signs of adenovirus-induced interstitial pneumonia in lungs. And that's a very explainable thing, because that's the site of adenovirus replication in humans. So, it correlated very well with analysis of viral copy numbers. So we could see viral replication in lungs. So after infection with both Ed5 and Ed5-3 virus, we could see it in spleen. And we were completely surprised not to see anything in the liver. And the histology exam didn't reveal any abnormalities or changes which could be related to adenovirus toxicity in the liver. And that was no unusual necropsy observation or macroscopic lesions in the liver. Once again, all these analyses were performed by the certified vet pathologist. So, I would like to remind you that 80% of viral particles administered systemically in a mouse model target the liver. And whatever we do, we can always track the adenoviral copy numbers in the urine liver, but we don't see it here. So, and the first study was conducted just to see if we can use the pig model for our Ed3 vectors. So we decided to repeat it. So, and we initiated another study, and thanks to Greg Billman's grant, so because those studies, in fact, are very expensive. So, and at this time we had more swine, more pigs. We have 3 pigs per group, and this study is still an ongoing study. And we are so anxious about liver toxicity that we decided to collect 9 different zones from the liver to analyze for adenovirus biodistribution and adenovirus-related toxicity. So, and of course, we also harvested other organs, 5 zones from the lungs, and we harvested heart and kidney and spleen and lymph nodes and pancreas. So, and at this time, we sacrificed the pigs. That's not we just took the liver biopsy. So we sacrificed the pigs 1 day after, 24 hours after, and then at other time points. So, and again, we don't see much adenovirus replication in the liver samples. So we can see something, and that's the 24-hour study in the spleen, so in the lungs, and almost nothing in the liver. So, and that's the staining of hexone. So, and I believe that Lisa tried her best to visualize, to find something in the liver, so but that's as much as we can get. So we can definitely see more in the lung tissues. And again, necropsy and histology analysis by vet pathologists demonstrate no abnormalities or changes that could be related to adenovirus toxicity. Blood and serum parameters are not affected. Body weights are normal, and so far we don't see any adenovirus-related toxicity in pigs. So what we have learned about the pigs? First of all, we learned that that's a very valuable and unique model to study performance of our viruses, not only at free retargeted, but in general. So all vectors which are based on adenovirus. And we also learned that biodistribution of adenovirus in pigs so much differs from what we see in mice. And we also learned that somehow, so adenovirus does not cause hepatotoxicity in pigs. But this study also raised a number of questions. And first of all, how much we can rely on virus distribution, safety, and efficacy data produced in mice. And secondly, which probably will have some resonance in the community. So is it reasonable to retarget our vectors and sacrifice their oncolytic potential by retargeting adenoviruses from the liver? As I explained, what we can do now, and that's what we do, we would modify the erythropoiesis, we would modify the adenoviral hexone and other structural components just to retarget from the liver. So thus, of course, sacrificing efficacy of adenoviral vectors and compromising the yield. But of course, we have to investigate these facts more and more. But in general, we are very much excited about this model, and we go further. And thanks to Lisa, now we can cultivate the primary swine cell lines. And here, so she managed to produce primary swine cells taken from our experimental pigs, 3 months old pigs. And she also produced a cell line from a newborn pig she borrowed from the farm. So, and it works very well. And now we do study our adenoviruses in those primary swine cell lines. So, and we do work with live tissues. So, and here, again, thanks to Elisa, so we can see that both swine pancreas and human PDAC tissues are positive for pancreatic marker cytokeratin 19, so we can use it in the future to target our viruses. And we are developing the protocol for non-invasive imaging of a pig pancreas. And thanks to Patrick Ballon, our collaborator from the Center for Magnetic Resonance Research, it turned out that pancreas in pigs localized so deep that we can't use the ultrasound-based imaging, and etc., etc. And of course, we are still interested in developing a pancreatic cancer model in swine. So, and we initially started this project as our collaboration with Recombinetics, but due to many reasons, Recombinetics no longer can continue working on this project, but we don't give up, and hopefully, and we can use a generic cancer model produced in National Resource and Research Center, for example, that will be just a P53 KRAS mutated cancer model, and we can we can activate P53 KRAS mutations with our adenovirus, which will be specific for pancreatic ductal cells. And of course, we need this model to study the efficacy and the safety of our therapeutics. And in general, the peak similarity to human anatomy and physiology and genetics make this model unique not only for virus-based therapies, but for pancreatic cancer research in general. So, and in the end, I would like to say that oncolytic virus therapy has witnessed both clinical achievements and failures, and it went through ups and downs, and very multiple ups and downs, and I believe that that's a very good time for virus therapy now. So we are not that much afraid of virus therapy, and the risk historically associated with virus therapy is lessening, and probably mainly because of all these great achievements in improvement of viral gene delivery, and of course, thanks to that fact that the very first oncolytic virus was recently approved by FDA to treat melanoma patients, and that's the herpes simplex virus. So, but it's not enough. I still believe that the biggest overall challenge facing the field is how to get these new viruses clinically tested. And the clinical testing of this virus is incredibly challenging. So because of this enormous amount of time and effort and funds required to bring these products to the phase 1 clinical trial. So on the other hand, there is nothing impossible, and if we really want, and that would be our ultimate goal, to bring this approach, especially this time we are focused on the NIS expressing adenovirus and the radioiodine-based therapy for pancreatic cancer to the clinical arena. And I would like to thank all these people for their tremendous support and help. Yes, and I have a long list of collaborators, and of course many thanks to Dr. Yamamoto and Lisa. Yes, you're here, yeah, but thank you very much, and I'm ready to take your questions.
I want to remind the people who are in other locations they can ask questions if they want, but we'll start here. Does anyone have a question or comment?
So in your introductory slides, you went over the history of the Onyx 015 virus, and you said that it's being used clinically in China right now. What's the basis upon which that was approved, and why isn't it being used in the U.S.?
Oh, yeah. I spent some time trying to understand the reason. So there are quite a few reasons. First of all, they didn't have this kind of stigma about adenovirus-based therapies at that time, as we had here after that Jess Gelsinger case. That's one of the reasons. Secondly, it's so much easier to recruit the patients in China rather than here. Thirdly, their clinical regulations are more loose than the clinical regulations we have here. For example, the patients are allowed to develop fever whenever we apply adenovirus-based therapy, which is a very normal thing. That's the normal environment how adenovirus works. It's not allowed here, and et cetera, and et cetera. So, but the main reason probably all this negative attitude in the end of 90s and in the beginning of 2000, which was formed around adenovirus-based therapy. It's getting so much better, but even right now we can see in reviews that now we feel sorry that we let this virus go. We do feel sorry.
Other questions or comments?
I'll follow up to Scott's question. Do they have side effects in China, or is that not reported, or is there a...
In terms of publications and report and statistic, we don't have much statistic. It's not kind of valuable, right? So, but this virus, the very same virus, was analyzed in phase 1 and phase 2 clinical trials here and in Europe. So, being administered via different routes, including systemic injections. So, and that was not effective. So, but it was as a monotherapy. But once again, so it was very well tolerated by cancer patients. So, but in China they treat mostly head and neck cancer.
I see.
So, and that's the intratumoral injection, and then you can go with a very high dose.
Sure.
So,
thank you. One other question. So you talked about being able to target these by putting different binding proteins on the surface, and you mentioned an RGD containing protein. So since I came from that area, part of the challenge with RGD is that it binds half a dozen integrins. So I would think that would off-target platelets and all sorts of other.
Exactly.
Is that an issue, or is it no longer used?
Integrins are highly expressed on the surface of many normal cells and tissues. That's a very known fact. But we made this modification just to improve efficacy and infectivity of viruses. So, but we control adenovirus replication via different mechanism. That virus is controlled with a tumor-specific promoter.
So related to the clinical trial things, I think still the challenge running the clinical trial here is a recruitment of a relatively early stage patients. In this country, actually, for example, this kind of gene therapy, the patient come to gene therapy is after failing 1 or 2 or 3 their pre-treatment, including clinical trials. Then after that coming here, in other countries, including China, you said actually probably the recruitment was a little bit earlier stage of the patients. First line, the second line level is possible. Do you think those kind of things affect our clinical trial?
Well, definitely. Yes, that's also one of the reasons. Yeah, it's more challenging to treat patients who join the clinical trials being diagnosed with phase 3 or 4. Yeah.
May I ask a question about, after modification with some of the CAR, this is the receptor, the endovirus can enter by this the receptor. So this one seems that your virus, endovirus seems, be modified but genetically, so there be CAR, the recognition to some of the other CAR will be losing. So do you think there are some receptor can be used for some modify some endovirus because they will be enhanced,
Well, definitely, that's a great question, and then that's the beauty of adenovirus because, as I mentioned, we know everything about adenovirus genome and the mechanism of its binding and interaction with the cells, and that's what, for example, Dr. Yamamoto is doing. But eventually, once you know the cell marker, you can retarget your virus So it would specifically bind to this, for example, a cell signature or the cell receptor. We can do it. So, and there are different mechanisms. And of course, here I demonstrated just a few, which are heavily used in my lab. But there are other mechanisms to retarget adenovirus from Coxsackie adenovirus receptor. So, yeah.
Thanks.
Any other questions or comments? All right. Thank you very much, Dr. Davydova.